All right, so we talked about the genetic code. Now here is the table for the genetic code. And because there are 4 bases, A, T, G, and C, or in the RNA actually, A, U, G, and C, right? A, U, G, and C. There are 4 possible bases in RNA. How many combinations of 3 can you have? Well, there's 4 for the first one, times 4 for the second letter in the code, times 4 in the third letter in the code. 4 times 4 times 4 is 64. So there's 64 possible 3-letter codes that you could come up with for RNA. When you're reading it 3 at a time, there's 64 possible things. But there's only 20 amino acids. In addition, there's also stop and start. You got to have some place to say, this is where you start making the protein, this is where you stop making the protein. So there's 2 more things that you need to deal with. But you're still only at 22. So what happens? There's duplication. If you notice here in serine, there's 4 different ways to say, we need a serine here. Wait a minute. There's 2 more over here. There's 6 ways to make serine. So in some cases, a minor mutation in the DNA might have 0 impact on the organism. If you have a DNA mutation that goes from UCU to UCC, the organism will not notice it one bit, probably. In advanced genetics, that's not maybe 100% true, but for the purposes of this, there's no difference. The protein that's going to be made is exactly the same. So there's lots of duplication. You can see that most of the proteins have at least 2 different ways to code for them. So how do we do it? The first base, you pick UCAG. Here's UCAG. Which is it? So if the first one is U, it could be anything in this first row. Anything in this first row. So there's a lot of choices. If the second one is C, well, it has to be in this first row. The second one is C, so now it must be in this box here. And then the third one is A. Here's A, reading in from the third base. UCA, there it is. Serine is the protein. If, on the other hand, I have the first letter in the codon is A, it must be this third row, something in this third row. The second letter is also A. Where does A intersect with A? Here in this box, right here. So it must be one of these 4. Now, the third base in the codon is G. Here's the G, AAG, it's lysine. The amino acid that goes there is lysine. All right. Now you can also see there's a couple of 3 different stop codons. Stop making the protein. The protein peptide is done. It's kicked out of the apparatus for manufacture. And whenever you hit a UAA, UAG, or UGA, that means stop. The job is done. Start, there's only one way. And this is all life. Starts at AUG. The universal code of life. This is everything. This isn't just plants. This is fungi and protists and you name it. AUG is methionine if it's in the middle, or it's also methionine start. It always starts at methionine. Now, when a protein is made, a peptide is made, it also gets processed later. That methionine might get chopped off before it's done being packaged and assembled. But the initial string, they always start with methionine. But you can have methionine in the middle as well. So if you're running along and you hit another AUG and you haven't hit a stop yet, just another methionine. And you keep adding more and more and more, you hit another AUG, another methionine. All right. So why do we need RNA2? There are 3 types of RNA. The messenger RNA is the only one we've talked about, that transcribed piece of RNA that copies a complementary strand of RNA from the original DNA template. That copy, that transcribed copy, is the message. It's the information from the DNA. The next type of RNA is called transfer RNA, which I'll show you in some next slides coming up. The transfer RNA actually is taking these amino acids and delivering them to the assembly line. And there are special pieces of RNA that are designed just to grab the right amino acid and deliver it. Grab the next amino acid, deliver it. And then the rRNA, or ribosomal RNA, is the RNA that is used as part of the actual protein assembly machinery, called a ribosome. And the ribosome is a mixture of protein and RNA. And it's this little molecular biological manufacturing unit, the ribosome, that does nothing except translate messenger RNA into protein. That's all it does. You can read the rest. Now, don't worry about introns and exons, other than it gets really complicated. Here's the DNA, and it's got all these things called introns and exons. What the hell does that mean? They're parts of the message that are actually thrown out before it gets turned into protein. It gets modified. So initially, you transcribe your DNA into RNA, and then all of these things called introns get removed. They get sliced out. They're thrown away. And here's my actual finished messenger RNA. A lot of people think the messenger RNA is an exact copy of the DNA, and it's not. It is highly modified in many cases, sometimes not, but it can be very modified and not the same. And once it's modified, that's your messenger RNA. I'm not going to get into introns and exons on the test. Again, this is more advanced concept, but you should know at the very least the simple idea that when DNA is transcribed into messenger RNA, the messenger RNA is not necessarily identical to the original gene, the original DNA strand. It could be modified. That much you should know. What about these transfer RNAs? This picture in green here is a drawing of an actual transfer RNA molecule. You can see it forms a very specific 3-dimensional shape with these pieces, one hanging off the end here. It's going to grab onto the appropriate amino acid. And because it has a 3-letter attachment site, the codon, it codes for the right amino acid. This attachment site, it technically isn't the codon, but it has the same concept. It's the same matching lock and key mechanism. It will only attach to a particular amino acid, either lysine or methionine or something else. It will only attach to one amino acid. And the anticodon is the portion that actually identifies it at the machinery, at the protein translation machinery called the ribosome. And when I show you this in the next slides, I hope this will make more sense. But these are lock and key mechanisms to make sure that only the right amino acid is being grabbed by this transfer RNA, and only the right amino acid is being delivered to the ribosome. The anticodon is the delivery key, and the amino acid attachment site is the literal lock and key to grab the right amino acid. Now you say, could there be 20 types of transfer RNA? Well, there's only 20 amino acids. Why would I need more? I need more because I need something for every one of those anticodons. Now, technically not 64 because at least 3 of them mean stop, right? So technically, I don't need those 3 to have a transfer RNA. I could do this with 61. But I need all the rest code for a protein, or excuse me, an amino acid. Every other of the 61 possibilities accounts for something. If you look at this table of 64 different things, all of them account for an amino acid. Every single one of these needs its own transfer RNA, with the exception of these 3 stops. Stops don't. They're not delivering an amino acid, so we don't need a transfer RNA for those 3. But the rest of them, the rest of the 64, 61 of them, we actually do need. All right, so here is the ribosomes. The ribosome is made up of different parts. These are protein and RNA parts, the large subunit, the small subunit. This is just like a machine that the messenger RNA feeds through, like reading 3 at a time in order to make protein. And we need ribosomal RNA to make this ribosome. It's made with protein and RNA, ribosomal RNA. So how does this work? So here is a messenger RNA. It goes to the ribosome and gets attached on the correct end and gets pulled through 3 at a time. So here you can see the codon on the messenger RNA attaching to the anti-codon of the transfer RNA. In green, we have the transfer RNA. And then it lines up 2 codons. The ribosome handles 2 codons at a time. One codon is just holding it in place, the one that went through prior. And then the next codon is the transfer RNA for the next amino acid going in. So that next amino acid lines up the transfer RNA, or excuse me, the next codon lines up, the transfer RNA joins. It already has the appropriate amino acid attached. And while it's held in this tight position, those 2 amino acids can be bound together. The ribosome will join those 2 amino acids together here, extending the chain. And then it clicks over one, and then the next one goes in and joins another amino acid, and then clicks over one. And it happens fast, hundreds per second. It's mind-boggling. So here you can see how the chain is elongating as I described. As soon as the new bond is made, the new amino acid is made, the old transfer RNA is kicked out to go pick up another amino acid. It goes to do more work, just sent for another load, and it clicks over to the next one, and round and round and round it goes. There are many videos on YouTube and things you could look at to understand this, but again, you don't have to understand in great detail. You need to understand messenger RNA, ribosomal RNA, transfer RNA, codons and anticodons, amino acids making peptide chains, which in turn are put together to make proteins. And this whole process on this screen is all about translation, because you're going to a whole new language. You're translating from the language of nucleic acids, the AUGC language, into these chains of 20 amino acids. So information is stored in the triplet codes of DNA, the codons in the DNA. This information is transcribed into 3 types of RNA. So mostly we talk about the messenger RNA, but where did that transfer RNA come from? Where did that ribosomal RNA come from? It also is coded in the DNA. Those other 2 types of RNA are also coded directly in the DNA. Messenger RNA carries information. It is transcribed information from the DNA. In the nucleus, introns are removed and the remaining exons spliced together to make functional messenger RNA. Transfer RNA attaches to specific amino acids. Ribosomal RNA and the proteins make up the ribosomes. Messenger RNA attaches to the ribosome and the message is decoded or translated. When the transfer RNA joins to the codon on the messenger RNA. Subsequent amino acids are attached to the growing peptide chain until a stop codon is reached. Just keeps going. It doesn't go to the end of the message. It doesn't go until it runs off the end. It goes until it hits the message stop. As soon as it hits stop, done. It's kicked off the ribosome, and the ribosome goes next, and another messenger RNA comes in. And these ribosomes, tens of thousands of these ribosomes outside of every nucleus and every cell are working factories constantly. I'm not going to go through this. You can walk through this, or you can find your own YouTube videos, or you can, whatever works. You just need to kind of get a feel for it and go, yeah, that makes sense. I get it. I get it. Without trying to overwhelm yourself with the specific names of any of the enzymes involved or anything else. What about a mutation? I think I better take a quick break.